Assalamualaikum. So today we'll talk about the ultraviolet visible spectrometer, or you can call UV-VIS spectrometer. What is it? It's a technique used to be measured the light absorbed across the ultraviolet and visible range of the electromagnetic spectrum. When incident light strikes a matter, it can either be absorbed, reflected, or transmitted. The absorption of radiation UV-VIS range causes atomic excitation, which refer to the transition of molecule from a low energy ground state to an excited state. So we will go on how the UV-VIS spectrometer works. First of all, we have the source of the light. At this case, we use lamp. We have presence of the light, then it passes through the magnetometer. When it reaches the prism, the second one, it will reflect into two rainbow color. But just one light will go to the next slit. Then it will striking the beam splitter and become two beam. Then the two beam will strike the detector, and the detector will be monitored by a computer.
My name is Iman and I will talk about the graphite furnace atomic absorption spectroscopy, or known as the FAAS. This instrument also has another name, which is the electrothermal atomic absorption spectroscopy, ETAAS. This is a type of spectrometry that is a known film method involving electrically heated graphite tubes. So DFAAS has been used primarily for analysis of low concentrations of metal in water samples such as arsenic, cadmium, mercury, lead, copper, chromium, and nickel. So what is the basic principle of this instrument? You can see here it utilizes the principle that elements in the gas phase absorb light at very specific wavelengths, which gives the technique's excellent specificity and detection limits. So here shows a diagram about 5 basic components in the FAAS. The first one is a source of light. For example, the source of light that we use is hollow cathode lamp. The function is to emit resonance line radiation. The second one is an atomization chamber, which is the graphite tube in which the sample is vaporized. So before the sample is placed on the flat form in the graphite tube, there are 3 essential steps happen in the graphite tube. First one is the drying step to remove solvent for sample. The second one is etching step to remove organic and inorganic material. And the third one is atomization step, the generation of free analyte atoms in light clouds. The third basic component of the FAAS is a monochromator. A monochromator is for selecting only one of the characteristic wavelengths, either visible or ultraviolet, of element interest. And the fourth one, a detector, which is a photomultiplier tube that measures the amount of absorption. And lastly, a signal process computer system, such as digital display or printer. That's all. Thank you.
Today, I'm going to explain about the inversely coupled plasma mass spectrometry (ICP-MS), which can detect the trace elements in the marine environment at very low concentration. So, let's see how the instrument functions. At first, the liquid sample has to introduce into the introduction sample system. Then it will get nebulized by the nebulizer. After that, it will enter into the spray chamber where it will generate fine aerosol. This fine aerosol will enter into the argon plasma. Each has temperature of 6000 to 8000 Kelvin. In this state, the sample will be ionized and atomized, and it will generate ions. The ions will be accepted into the interface region, where it consists of sample cone and skimmer cone. After that, the ions will be entered into the ion optics. The ion optics will focus and guide the ion beam into the mass quadruple analyzer. In this state, the mass quadruple analyzer will separate the ions according to the mass charge ratio, and then the ion will be measured at the detector. The ICP-MS can detect several types of trace elements, for example, zinc, copper, ferrum, vanadium, and so on in the marine environment.
Okay, the instrument that I choose is Energy Dispensing X-ray Fluorescence, or EDXRF. So, how it works? Energy dispensing X-ray fluorescence is suitable for determination of matter in marine sediment. EDXRF is designed to analyze group of elements simultaneously. This type of XRF instrumentation separate the characteristic X-ray of different elements into a complete fluorescent energy spectrum, which is processed for qualitative and quantitative analysis. Filter position between sample and detector are used to improve signal, background reduction, and focus on certain region of the spectra. Next, for fundamental principle, an alternative process is indirect excitation, in which the observed fluorescence is produced as secondary process by photon. X-ray is an electromagnetic radiation generated by high-energy particle atom. This radiation has wave-particle duality. X-ray fluorescence XRF use primary X-ray photon to excite the atom to produce secondary XRF for material composition analysis. Okay, the last one, type of pollutant that can be analyzed is heavy metal pollution assessment, which are ferrum, chromium, cuprum, and zinc. Mostly it's found at soil in the vicinity of industrial area. It's because the soil near a pulp and paper mill was found to be highly polluted by heavy metal.
Okay, so the next instrument is Fourier transform infrared spectroscopy, or simply known as FTIR spectroscopy. This instrument can be used to identify microplastic. What is microplastic? A microplastic is a small piece of plastic that the size is less than 5 mm, which come from various of sources such as wrappers and bottle caps. Abundance of microplastic can cause several harmful physical effects on humans and other living organisms through such mechanisms as entanglement and injection. Okay, let's go back to our main topic, which is FTIR spectroscopy. FTIR is a technology that can identify chemicals by using infrared light source to measure their absorption. In FTIR spectroscopy, the sample will be exposed to different wavelengths of infrared light. An interference is produced from two beams that are separated by a beam splitter. The detected interference waveform is called Fourier transform, and the spectrum is obtained by separating each wave number. Then, the spectrum obtained can be compared with known spectra to find its match. I think that's all I can conclude based on my finding about FTIR spectroscopy.
Hi, Assalamualaikum. Okay, let's move to the next instrument, which is voltammetric in situ profiling system, or known as VIP system. Okay, you can look at the picture given there. This is the VIP system. Okay, this VIP system is for continuous real-time monitoring of trace metals such as copper, plumber, cadmium, and zinc. Okay, this VIP system can be used at both fresh and seawater. Okay, the VIP system consists of voltammetric probe with internal hardware that also houses a voltammetric cell through which sample is passed using a peristaltic pump. Okay, the voltammetric probe is remotely operated from the surface via a laptop by means of a coaxial cable that enables data transmission. Okay, the heart of the submersible voltammetric probe is a HG-plated IR based microsensor covered with an anti-fouling gel membrane. What is this gel functioning? Okay, the gel is play an important role in this system. Okay, this gel integrated microsensor allow direct voltammetric measurement of trace metal in complex media without physical and chemical interference of the test solution. Okay, so how this VIP system work? Okay, the sample is pumped through the cell and trace metal are reduced and pre-centered or deposited in the HG film. After deposition, usually between 5 to 30 minutes, the metal are reoxidized at the specific reduction potential, producing an anodic current directly proportional to the concentration of trace metal in the sample. Okay, that's how it works. Okay, so this VAP system is robust and reliable because of this in situ measurement of mobile fraction of trace metal can be achieved down to a very low concentration level. Okay, that's it.